Hello everyone, Immunology Explained here, and today I'm bringing you a brief video which will explain the concept of the Enzyme-Linked Immunosorbent Assay, or as it's more commonly known as, the ELISA. Now, the ELISA is an extremely commonly used lab technique all across the world due to its high sensitivity, relative ease to set up and carry out, and most importantly, due to its customizability. As you will see when I get into describing the components of the ELISA, you can swap in different reagents to customize the ELISA to target almost any molecule or protein of interest. Just a brief disclaimer, in this video I will be describing the classic sandwich ELISA. There are many variations of the ELISA, but many of them follow the same steps. Without further ado, let's get started. So in order to understand how ELISAs work, we need to again briefly touch on the immune system. Antibodies are proteins produced by B cells, which circulate throughout our body and are capable of binding to foreign substances. They do this through highly diverse binding sites located on their two variable regions. Antibodies also have constant regions which are used for carrying out effector functions of the immune system. With these binding regions, antibodies bind to antigens, which can be broadly categorized as foreign substances. This can include components of bacteria, viruses, or even harmless substances such as eggs or peanuts, which results in allergies. ELISAs take advantage of this property of the immune system, the specific binding capabilities of antibodies. As I mentioned in the introduction, ELISAs have many applications, which is partially what makes it such a widely used technique. ELISAs are often used as medical diagnostic tools for detecting serum antibody levels or concentrations in patient samples. They can also be used to detect food allergen contaminants. They're commonly used to detect drug levels. And again, just to reiterate, the major strength of an ELISA is its ability to identify and quantify unknown substances in a sample if you have the right reagents. Onto the reagents, I'll now describe the main components of an ELISA before describing exactly how an ELISA works. The first component of an ELISA is the capture antibody. This is what is used to typically coat the plate with, and functions to capture your molecule of interest. The second component is the sandwich antibody. This antibody is typically specific for the same protein as the capture antibody, and serves to form a binding site for the detection antibody. The detection antibody is conjugated to an enzyme which plays a key role in ELISA detection, as will be explained. In some cases, a single antibody can function as both the sandwich and detection antibody if it is conjugated to an enzyme and specific for your protein of interest. Next is your solution of interest, which contains an unknown level of your molecule which you're studying. The next component is the substrate. The substrate also plays a key role in the detection step of an ELISA. The product is the final readout of an ELISA. It typically manifests as a change of color of the solution of target wells. This change in color can be quantified by a plate reader, which is capable of detecting altered light absorbance as the solution changes color. The next component is blocking protein. This protein plays a key role in maintaining specificity and reducing background in an ELISA. And finally, there's the plate itself. You can use a variety of different plates to carry out an ELISA, but the most common format is a standard 96 well plate. Now let's get into the specific steps of an ELISA. So I'm going to describe what happens in each individual well of your 96 well ELISA plate. The first step of any ELISA is adding the coding antibody. So again, this is the capture antibody. This serves to capture your protein of interest. This step is typically done by adding a solution containing your capture antibody to each well and leaving the plate overnight at 4 degrees Celsius. In some cases, you can buy plates that are pre-coated with your capture antibody. In this case, you can move directly on to step 2, which is the blocking step. Blocking is a very important step because it serves to prevent the binding of any other molecules or non-specific antibodies to the plate. This reduces background and helps to get cleaner results. Blocking solution typically consists of PBS or saline, consisting of 2 to 10% bovine or calf serum albumin. The third step is adding your solution of interest. This allows the molecule in the solution to interact with the capture antibody in each well. To enhance these interactions, ELISA plates are often placed onto plate shakers and continually mixed. The next step is adding the sandwich antibody, which again serves as a scaffold for detection. Finally, the detection antibody is added. It binds to the constant region of the sandwich antibody, ensuring that enzymatic activity only remains in wells where your molecule of interest has been captured by the sandwich. The final step in an ELISA is adding the substrate. The substrate you use depends on what type of enzyme is conjugated to your detection antibody. In many cases, this enzyme is a phosphatase or horseradish peroxidase. These enzymes will react with your substrate, causing it to change the color of the solution. This color change occurs only when there has been sufficient capture of your molecule of interest. As previously stated, this color change can be quantified by a plate reader, which measures the absorbance of light in each well. To conclude, I'd like to go over some general tips to remember when carrying out an ELISA. Firstly, it is very important to have the proper controls when conducting an ELISA. You should have wells that function as positive and negative controls to ensure that your assay is working, as well as a standard if it is applicable to your protein of interest. When performing washes between each step, it is important to get as much of the fluid out of each well as possible. This can be done by quickly inverting the plate over a sink in a process known as flicking. The plate can then be tapped on paper towel several times to dry each well for the next step. Finally, most ELISA plates have numerical and alphabetical labels. It's important to utilize these labels to keep track of where your samples are on the plate and to avoid adding something to the wrong well. This also makes your analysis much easier. Here's an example of an ELISA I ran in my lab. As you can see, the wells which have turned yellow have meaningful antigen capture, whereas the wells that remain blank did not. Thanks for watching. If you enjoyed this video and learned something, please make sure to leave a like, and subscribe for more future immunology related content.